In this presentation, I will be discussing how immunohistochemistry could be used to identify the primary tumour. In the liver, you have liver lobules, which contain 6 portal triads that are made up of a hepatic duct, hepatic portal vein, and a hepatic artery. These 6 portal triads surround one central vein. In this structure, you have the hepatic phagocytic cells, hepatocytes, hepatic sinusoids, common bile ducts, hepatic cords, and more. Viewing one section of the core liver biopsy with an objective lens of 10, we are able to see sections of steatosis. However, this is not the primary concern, but we are able to view plates of hepatocytes with sinusoids running between them. In Figure 2, we are able to see necrotic tissue surrounded by darker stained cells, which are the tumour cells. This is known as dirty necrosis. Close to this, we are able to view, at an objective lens of 5, more tumour cells with surrounding fibrous tissue as a response to the tumour. It is important that we are able to diagnose the primary cancer. Here the tumour cells are not as well differentiated as they are in the colon, and hepatic cancers are not glandular. It is safe to assume that this is not a primary hepatic cancer. However, this may be a cholangiocarcinoma, also known as a bile duct cancer. Cholangiocarcinomas are gland-forming tumours. Keratin markers could be used to identify if this is a primary colorectal carcinoma or a cholangiocarcinoma. The appendix is found in the gut that typically expresses keratin 20, which could be used as a positive control. Using an objective lens of 2, we can see that the epithelium facing the lumen of the appendix is positive for keratin 20. The liver core biopsy beside it, seen in Figure 4 using a magnification of 4.5, contains tumour cells which are positive for keratin 20, but the hepatocytes are negative for keratin 20. This suggests that the tumour cells could be derived from the gut. Keratin 7 is usually associated with cholangiocarcinoma, which is normally expressed in the epithelium of tonsils. Here, a lymph node has been used as a positive control, which stained positive for keratin 7. Beside this is the liver core biopsy seen in Figure 6 using objective lens of 5, which is negative for keratin 7. However, the bile duct cells stain positive for keratin 7, but the tumour cells stain negative for keratin 7. From this immunohistochemistry staining, we can assume that the cancer cells are derived from the gut, as they stain positive for keratin 20, but negative for keratin 7. Therefore, we can assume that the primary tumour is colorectal carcinoma.